Hello everyone, I hope you're doing well during these times. My name is Alexis Peretto, and today I'm going to present the postmortem interval determination using microbes, and my mentor was Professor Zarati. So to begin, let's talk about the microbiome. The microbiome is composed of many different microorganisms, and these microorganisms are present pre and postmortem. But postmortem, these microorganisms inhabit the body and allow decomposition to occur. Um, and there's a variety of microorganisms that exist, and they depend on body site and the stage of decomposition, but there is a great amount of them present. These microbes can be taken into account forensically when determining the postmortem interval, and specifically, scientists are interested in looking at the necrobiome and the phantom microbiome, which is considered the microbiome postmortem or after death, and they contain microorganisms such as bacteria, arthropods, and fungi. So microbiome samples. There's two different ways to obtain microbiome samples, and these include culture-dependent methods and culture-independent methods. And culture-dependent is collecting a sample of microbes and isolating certain microbes and then sequencing them to identify them, where culture-independent is just taking the sample and sequencing all the microbes present and taxonomically identifying them. To collect these samples, they use a sterile cotton applicator, and they swab this over the site of interest, and then they preserve it by putting it back into a tube, such as a capture tube. So procedures that are used are stereomicroscope, which they look at the microorganisms under the microscope, and they're able to taxonomically identify them that way. Three other ways are DNA barcoding, qPCR, and 16S rRNA, and here they extract and amplify the DNA of the microorganisms to sequence them in order to identify them. Once they have been sequenced, they are placed into an analyzation database, such as machine learning, which uses the three databases known as ExoBoost, Random Forest Regression, and Neural Network. So let's talk about the microbiome's involvement in the postmortem. So microbe communities during decomposition vary depending on the stage of decomposition, as well as the time of year. Their diversity may increase or decrease, or there may be different types of microorganisms that exist. So when researchers are doing their research, they either use human cadavers or animal cadavers. A lot of the research that I found, they use human cadavers, but there are a lot that also use animal cadavers, such as pigs and rats and mice of sorts. So it is important to remember that throughout decomposition, there is a variety of microbes, and these microbes will change during the decomposition process. And those changes that occur of microbes can help aid in the determination of the postmortem interval. Also, it is important to remember that decomposition does occur in water as well. It may be different, but there are microorganisms that are present, and these microorganisms create what is known as a biofilm. And this biofilm contains a variety of microorganisms, but these microorganisms are epikenotic communities. And these communities contain microbes that are very important when determining the postmortem interval. They contain a lot of information, and they help create a more accurate postmortem interval. Also, something to remember is that microorganisms transmigrate, meaning they move throughout the body, they move into the soil. So they have the ability to move and help aid in decomposition of different sites. So because of their transmigration, soil samples can also be used and collected to look at the microbes present. And these microbes can be taxonomically identified, or we can look at the community at which they belong, and this will help determine maybe which stage of decomposition the body is in. Also, there's a presence of microflora, and these microflora are definitely something that, when present, we want to take into account when determining the postmortem interval. So scientists or researchers have looked at specific microbiome sites, such as the oral cavity or the gut. The oral cavity contains a lot of proteobacteria, and these proteobacteria are present throughout all the stages of decomposition. So they're definitely something that is being taken into account when determining the postmortem interval. But it was also determined that looking at the oral microbes that exist throughout decomposition, that as time and decomposition increases, the diversity decreases. So there's a limited amount of microbes that are present to help aid in the postmortem determination further on. But there is a microbial change over time. The gut microbiome stated that during the bloat phase, which is earlier on in decomposition, there's a great diversity of microorganisms, and all those microorganisms, the variety of them is very useful when determining the postmortem interval. But again, as time goes on, there is a change and a lack of in diversity of microorganisms occur, which will change the way we look at them, but whatever is there will help determine the postmortem interval, but earlier on is more useful. Bacterial dials were determined to be the best estimators of the postmortem interval when they are present. So machine learning is defined as a new type of learning implementation, which is done on computers, and it uses different computer functions to help determine the postmortem interval. So there were three, I believe, machine learning approaches. This one is with the postmortem skin, and they looked at the nose and ear microbes. And so when they looked at they collected the samples and they did the 16S rRNA sequencing on them. And when they placed it into machine learning, it was able to calculate the microbial diversity and analyze this data that they obtained from these samples. And a postmortem interval of plus or minus 2 days was created by this machine learning approach. And so meaning that plus or minus 2 days is the postmortem interval predicted could be 2 days earlier or 2 days later, but that is the plus or minus 2 days is great considering its accuracy. So the other ones they did were just microbial samples and postmortem microbes. So the microbial samples were collected from the skin, such as the torso and the head, as well as the grave soil. And they used random forest regressor for this. And it was indicated that, as I said before, with like the gut and the oral cavity, the microbes that are present during the early stages of decomposition are hold great value, and the ones that are collected early in this case and put into random forest regressor are the ones that created a better post-mortem interval determination compared to the later on microbes. The other one was just the post-mortem microbes, and this had used metadata as well as samples collected from the eyes, ears, nose, mouth, and rectum from the Wayne County Medical Examiner in Detroit. Those were collected, and placing that data and the metadata into random forest neural network as well as ExoBoost, it was determined that ExoBoost created the most accurate postmortem interval. So when implementing this, ExoBoost has a higher value in determining a more accurate postmortem interval, though random forest and neural network were not far behind. As always, there are challenges and limitations to new things that we like to implement, such as the microbes. And this is because there are variant microorganisms, meaning there's many different kinds, but some that have not been taxonomically identified, which can pose a problem because we can't identify them, so they aren't useful to us in that sense. Collection and preservation is very general, so that's a limitation because as always, we want to ensure that our samples are collected and to the best way they can be and preserved. Highly dimensional data, as well as the understanding of the usage of microbes, such as navigating and understanding it in court. We want to ensure that everyone understands how useful they are, what they do, what they mean when it comes to determining the postmortem interval, and how their implementation can aid in determining a more accurate postmortem interval. It has also determined an invasion of privacy because you're looking at someone's personal microbiome. Future research is being conducted to ensure we fill in the gaps, such as the production and usage or the proper implementation to ensure that the microbes are being used correctly and implemented correctly to be able to be used to determine the postmortem interval. Standard protocols to ensure that we store them or package them and preserve them correctly to ensure we don't get erroneous results or there is any contamination. Also, future research to make sure that a more accurate postmortem interval can be determined, that we're using all the microbes to the best of our abilities, and that we're using the data right, and those kinds of things. Postmortem interval determination with the microbes, the takeaway is that these microbes can hold a lot of information. They tell us the stages of decomposition, and they hold just a lot of information that can be useful once they're taxonomically identified. And it helps us determine the stages, as I said. So it's highly effective in aiding forensic scientists when determining the postmortem interval. And that's something that we have to look forward to because it's helping us be more accurate and give a better picture. And using this also gives us a better timeline of what happened up to the point where the body began to begin decomposition. And that is very, very important. So thank you for listening, and here are my references.